Hi, my name is Hwang Lee, a doctor's course student in the Systems Biology Laboratory. Today, I'll give you a presentation on the later part of the Chapter 11 of our textbook, the therapeutic application of gonadotrophins. For the outline, I'll first introduce about what the gonadotrophins are and their molecular structure and function. Then we will discuss about the medical and veterinary application of them and the recombinant hormone recently approved. The gonadotrophins are a family of hormones for which the gonads represent their primary target. As you can see from the figure, most gonadotrophins are synthesized by the pituitary, and some of them are made by reproductive and associated tissues. They directly and indirectly regulate reproductive function and the development of secondary sexual characteristics. Here are the three gonadotrophins we will study in this lecture. FSH and LH are mainly known as regulators of female reproductive cycle. And HCG is the hormone used in pregnancy test together with LH. In this chapter, we will study the basic structure and function of these hormones. First, for the molecular structure of these hormones, as you can see on the figure, all these hormones are heterodimers containing an identical alpha subunit and unique beta subunit for each of them. So the beta subunit confers the biological specificity to each of the gonadotrophins. First is the molecular structure of the FSH. Human FSH displays 4 asparagine-linked glycosylation sites. The oligosaccharide components attached to them play a direct and central role in the biosynthesis, secretion, and potency of the gonadotrophins. The sugar components attached to the alpha subunit play an important role in dimer assembly and stability, as well as hormone secretion and possibly signal transduction. While the sugars associated with the beta subunit also contribute to dimer assembly and secretion, they appear to play a more prominent role in the clearance of the hormone from circulation. Next is the structure of LH and HCG. The gene coding for the beta subunit of LH exhibits significant amino acid homology to HCG. So both hormones act via the same receptor and promote similar biological effect. According to an article published last year, they act on the same receptor but result in quantitatively and qualitatively different effect. But overall, we'll focus on the similar roles of them in this lecture. Now, I will talk about the function of FSH and LH. Even though they are named for what they do for the ovaries of the female, in fact, FSH and LH affect the reproductive organs of both the females and the male. In male, the major target of FSH is the salutary cells found in the walls of semi-inferous tubules of the testis, and it stimulates sperm production. On the other hand, LH promotes synthesis of testosterone by the testicular Leydig cells. In the female, FSH mainly targets the granulosa cells of the ovarian follicle. FSH stimulates their division, thus follicular growth and development. On the other hand, LH mainly targets the thicker cells and plays a major role in maturation of the dominant follicle and ovulation. This is the summary table of function of the FSH and LH. Next, PMSG is a unique member of a gonadotrophin family of hormones. It is synthesized only by pregnant female horses and not found in any other species. Also, it displays both FSH-like and LH-like biological activities. PMSG is secreted by cup-shaped outgrowth found in the horns of uterus of pregnant horses. They synthesize high levels of PMSG and secrete it into the blood, where it is detectable between days 40 and 130 of gestation. The hormone is commonly used to induce ovulation in livestock prior to artificial insemination. The inhibin and activins are a family of dimeric growth factors synthesized in the gonads. The inhibins are heterodimers consisting of alpha and beta subunits, and activins are beta-beta dimers. Two closely related but structurally distinct beta subunit forms have been characterized as beta A and beta B. The naming and polypeptide composition of the inhibin-activin family may be summarized in the left figure. Inhibins and activins were initially identified as gonadal-derived proteins capable of inhibiting or stimulating pituitary FSH production. However, they also likely induce responses in tissues other than the pituitary and gonads. For example, a recent study involving inhibin-deficient transgenic mice revealed a novel role for inhibin as a tumor suppressor. Because of their central role in maintaining reproductive function, the gonadotrophins have therapeutic potential in treating subfertility in human and animals. The market for these hormones is substantial. By the late 1990s, annual human market stood at about $250 million. Although the human pituitary is the obvious source of human gonadotrophins, we need much more to use them in clinical purposes, and the urine of post-menopausal women can be used for these purposes because they have both FSH and LH activities. 2.5 liters of urine may be required to produce 1 dose of human FSH. And the procedure by which these hormones can be purified is described in the flow chart on this slide. Menotrophin is FSH-enriched extract from human urine. It is administered to stimulate follicular maturation, with subsequent administration of HCG to promote ovulation and corpus formation. Altogether, they lead to fertilization of the infertile subject. They are also used in the treatment of men's subfertility or related conditions. Gonadotrophins are now also produced by recombinant DNA technology. Recombinant human FSH produced in CHO cells has proven clinically effective. When administered to humans, it yields no unexpected side effects and does not show immunological responses. RHFSH has proven to stimulate follicular growth in females. It was the first biopharmaceutical substance to be approved for general medical use in Europe. Recombinant gonadotrophins approved for general medical use are listed in the table on this slide. Gonadotrophins may be utilized to treat subfertility in animals and are routinely used to induce superovulatory responses in valuable animals such as horses and cattle. The theory and practice of superovulation is quite similar to the use of gonadotrophins to assist in vitro fertilization procedure in humans. Exogenous FSH is administered to the animal so that multiple follicles can develop simultaneously. After administration of LH to help promote ovulation, the animal is mated to fertilize the released egg cells, and then some viable embryos may be produced, typically from 4 to 10. Then the embryos are recovered from the animal and maintained in cell culture for a short period of time. A single embryo is then re-implanted into the donor female, while the remaining embryos are implanted into other recipient animals who act as surrogate mothers. This technology is most often applied to valuable animals such as prize-winning horses or high milk yield dairy cattle in order to boost their effective reproductive capacity several-fold. For human drugs, 3 additional recombinant hormones have recently gained marketing approval: thyroid-stimulating hormone, parathyroid hormone, and calcitonin. The commercial recombinant TSH product, Thyrogen, is produced in a CHO cell line with plasmid harboring the DNA sequences coding for the alpha and beta TSH subunit. When administered to a subject, it shows radioactive responses with iodine in the reoccurring active thyroid cancer cell. So TSH is approved for medical use as a diagnostic aid in the detection of thyroid cancer. On the other hand, parathyroid hormones function as a primary regulator of calcium and phosphate metabolism in bones. It is produced in E. coli and purified and presented as a sterile solution. So the commercial product, Forteo, has been approved for the treatment of osteoporosis in postmenopausal women. Lastly, calcitonin is a hormone which plays a central role in regulating serum ionized calcium and inorganic phosphate levels. It is approved to use clinically to treat hypocalcemia associated with some forms of malignancy. In conclusion, several hormone preparations have a long history of use as therapeutic agents. In virtually all instances, they are administered simply to compensate for lower endogenous production of a hormone than the normal level. As a result, gonadotrophins have allowed hundreds of thousands of subfertile individuals to conceive. Although most such hormones were in medical use prior to the advent of genetic engineering, however, the recombinant hormonal preparations are now gaining greater favor, mainly on safety grounds. So hormone therapy will remain a central therapeutic tool for clinicians for many years to come. Thank you for listening.